For those of you who weren't in attendance yesterday, let me just make a couple of comments. We will have 3 speakers in the session that will be connected at the same time. And then every attendant had the possibility to make some questions to any of the speakers on the platform by writing. I will receive the questions in here, and after the third talk, we will have a 15 minutes discussion where I can pass the questions along to the corresponding speaker. Regarding the talks, I just remind the speakers that you have each 25 minutes. Please try to stick to this time to avoid any delays in the session. And when the other speakers are talking, please just keep your microphone in mute to avoid interference. Having said that, we're gonna then kick off the session. And our first speaker of the day will be Dr. Adolfo López de Munain from the Biodonostia Research Institute in Donostia. Adolfo, you have the floor. Please go ahead.
Early onset of the disease being more severe in neonatal forms and with years of evolution, as you can see in the picture. The multisystemic degeneration includes, moreover than progressive muscular weakness, atrophy, and myotonia, the clinical typical traits of myotonic dystrophy, some other traits such as cardiomyopathy, insulin resistance, cataracts, increased cancer incidence, dementia with tau protein deposits in the brain, metabolic syndrome, and premature death, being respiratory failure, and the heart arrhythmias, the most frequent cause of death. Taken together, all of these events associated to aging occur prematurely in DM1. At the molecular level, the CTG expansion in DMPK gene has several effects, with evidence that all of them participates in the pathophysiology of DM1. The CTG repeat expansion reduces the DMPK transcription, reduce also the transcription of neighboring genes like CIS1, CIS5, which contributes to some DEM traits such cataracts. And the third mechanism probably is the most important. With this mechanism, mutant DMPK transcripts accumulate in ribonuclear foci that interfere with 2 families of proteins that regulate the splicing, muscle-blind and COP, that both interferes with RNA splicing of multiple genes. Different pathogenic mechanisms, including alteration of autophagy, mitochondrial dysfunction, senescence accumulation, and telomere shortening, and genomic instability, have been associated with DM phenotypes. Most of these mechanisms have been reported in muscle satellite cells, and taken together permit us consider DM1 as a progeroid disease because it recapitulates most of the clinical hallmarks of aging. In order to characterize the underlying links between the pathogenic mechanism of DM1 and aging phenomena, we developed the following task. First, we performed transcriptomic analysis of primary fibroblasts derived from healthy donors and DM1 patients. Then we validated these results in patient-derived myoblasts and peripheral blood mononuclear cells. And finally, we validate the results in DMXXL mouse, as well as Drosophila models of the disease. And finally, we test the capacity to recover phenotype of senolytic compounds, quercetin, dasatinib, and navitoclast. So we established fibroblast culture from 7 different DM1 patients and 5 healthy donors. First, we determined whether fibroblasts recapitulated the molecular alteration observed in DM1 patients with Western blot, immunofluorescence, and qPCR, showed that the expression of these genes, DMPK, muscle-blind and 6-5, was reduced at mRNA level, whereas curve was elevated in DM1 derived fibroblasts. In addition, we detect the accumulation of nuclear foci and the expression of the neonatal isoform of muscle-blind 2 with inclusion of exon 7 in DM1 fibroblasts. We also show the correlation between the severity of molecular alteration with the number of CTG expansion in the upper part of the slide and with the muscle impairment rating scale, also named Mills scale scores. These results indicate that patient-derived fibroblasts maintain the altered molecular and genetic patterns of DM1 patients. At transcriptomic level, we identify in these fibroblasts 378 genes that were significantly altered in DM1 fibroblasts. 292 are down-regulated and 86 are up-regulated. Gene ontology analysis classified these genes as genes related to cell cycle, cell division and replication, and DDR, DNA damage response, where the processes more significantly altered in DM1 derived fibroblasts, all of which are down-regulated. Next, we validate the transcriptomic alteration in additional samples of DM1 patients. Interestingly, DM1 fibroblasts displayed lower expression levels of genes involved in cell cycle and some others involved in cell division and DDR, among other genes. We also studied these pathways in DM1 PBMC, primary human DM1 myoblast and myoblasts from tibial anterior of DMXL mice, the mice model of DM1. And we found that the same results as in fibroblasts. Correlation analysis confirmed significant positive association between muscle-blind and DMPK1 and between them and cell cycle genes. Also, we found the same alteration in myoblast and in DMSXL myoblast, suggesting a central role of cellular proliferation in DM1 pathogenesis. Well, we also found a significant decrease in the growth of DM1-derived fibroblasts compared with control. This was accompanied by a remarkable reduction of the number of cells positive for phosphohistone H3, a well-established marker of mitosis and cell division. Next, we investigate whether the accumulated deregulation of cell cycle, replication, division, and DDR could trigger the acquisition of a permanent senescent state in DM1. We performed a several passage protocol, which is a well-established methodology to study senescence in vitro, and counted the number of population doublings after approximately 40 passages. DM1 fibroblasts show a decreased division rate compared with control cells, and interestingly, they also require a lower number of division and passages to undergo senescence and stop dividing, and this was accompanied by the presence of higher number of senescence-associated beta-galactosidase positive DM1 fibroblasts. We studied P16, P14, P21, and P27 expression, and we found that the levels increased at late passages with higher levels of in DM1 fibroblasts than in control cells. Levels of BMI1 is a member of a polycomb family with a central role in cell proliferation through the repression of P16, P14, and P21 were also lower in DM1 cells. Then we studied these cell cycle regulators P16, P14, P21, and P27 and BMI1 in 3 additional cellular models of DM1 in primary fibroblast, in PBMC, and DMXL mice. And we found the same alteration than DM fibroblasts, suggesting a central role of cellular proliferation in DM1 pathogenesis. To determine whether BMI1 was involved in the reduced proliferative capacity of DM1 cells, we overexpressed it in control and in DM1 fibroblasts via lentiviral infection. After confirming the increase in BMI1 expression at the protein and RNA level, we performed functional studies and found that BMI1 restoration rescued the impaired cell growth and proliferation of the DM1 fibroblasts, similar to a similar extent to the control cells, as you can see in this slide. In parallel, DM1 fibroblasts overexpressing BMI1 and also a significantly reduced levels of P16, P14, P20, P21. Moreover, we also found that BMI1 overexpression decreased the number of sub-beta-gal positive cells. Next, we analyzed the secretory profiling of DM1 cells, and we found that the main pro-inflammatory cytokines are increased more in late passages in DM1 patients, again controls, and especially interleukin-6. Xenotherapy is a novel therapeutic strategy that aims to permanently remove senescent cells. Given the premature and enhanced senescence of the DM cells, as you can see in the previous slide, we investigated the efficacy of this type of therapy against DM1. We treat late passages DM1 and control fibroblasts with 3 independent senolytic compounds, quercetin, dasatinib, and navitoclax during 72 hours. First, we quantified the beta-gal staining and observed a dose-dependent decrease in the number of positive cells in control and DM1 fibroblasts after treatment with each of the 3 senolytic compounds independently. We performed similar experiments in early passage fibroblasts. In this context, senolytic treatment also decreased the number of beta-gal positive cells and increased the number of caspase-3 positive cells, similar to a similar extent in control and DM1 cells. Next, we validated these in vitro results, with in vitro results in a Drosophila model of DM1 that expressed 480 interrupt CTG repeats. This fly was given very kindly by Professor Artero from the University of Valencia. First, we found that DM1 flies present impaired locomotor activity and a reduced lifespan, a very strong reduction of lifespan compared with 2 control flies lacking CTG repeat, indicative of an accelerated aging phenotype. In line with this idea, the expression of markers of senescence with homologs of P21 and P27, the couple, and PCC homolog of BMI1 was elevated and reduced respectively in DM flies. In this context, DM1 flies show reduced levels of muscle, string, seed, and canna, further validating the relevance of results obtained in the transcriptomic study. Interestingly, 5 micromolar quercetin rescued the expression of muscle blind, string, seed, and canna, And similarly, the levels of DACAPO and PCC were restored after the treatment with quercetin. Overall, these results confirm the senolytic treatment recovers molecular alteration in DM1, also in vivo. Finally,